Hey guys. Hey, look, I figured out how to have myself there in the bottom so you can see me. Hey. Yay, technology. Okay, so anyway, we're going to go over control of gene expression this week. So how we got our genes, we went over last week. Now, how do we turn them on, turn them off, all those kinds of things. Okay, so the big questions are how are genes turned on and off in eukaryotes? And how do cells with the same genes differentiate to perform completely different specialized functions? Okay, because when we start out as babies, we're just like a little ball of cells, right? So we start out with two cells. We have our egg cell and then our sperm cell. How do we become the being that we are today? How does our skin cells know to become our skin cells? How do our heart cells know to become our heart cells? Things like that. So there's kind of the evolutionary background of it. So for prokaryotes, obviously they're single-celled. They evolve to grow and divide very quickly because they have to respond to changes in their external environment a lot more and a lot quicker. For gene regulation, how you have to evolve kind of like how to turn genes on and off quickly, and that way they can be flexible and reversible. And so they have to adjust the levels of enzymes for synthesis, for making those genes, and digestion for breaking them down. So for eukaryotes, we are obviously multicellular. We evolved to maintain constant internal conditions, so homeostasis, while our external conditions are changing. So we have to regulate the body as a whole in terms of growth and development. And because of that, it's a very long-term process. So you have specialization happening, which is turning on and off large numbers of genes. Specialization is like telling your cells that they have to be specific cells. So you have to coordinate with your entire body rather than serving the needs of each individual cell, okay? Because all of the cells that you have are eventually going to grow to become, different specialized cells. So in humans, we have something called genetic totipotency. Okay, so that means all the cells except your gametes, okay, have the same genetic information. So every single bit of your cells except your sex cells have the same genetic information. In total, human eukaryotic cells have about 35,000 genes. Genes are expressed in all cells all the time. Okay, you have specific housekeeping genes that maintain certain housekeeping aspects for your cells. Some genes are expressed after the cell becomes differentiated. So say our skin cell becomes a skin cell, and once it is differentiated into a skin cell, then some genes can be expressed after that. Some genes are expressed all the time in specific cells, and some are expressed only as conditions around the cell changes. So here are the levels of control. So the control of gene expression can occur at any step in the pathway from the gene to the functional protein, okay? So starting out from the genetic code from your DNA all the way to becoming a protein. So these are kind of the main steps for the levels of control. So you can have control at packing or unpacking the DNA. Okay, so when we kind of open it up so that way we can get ready to go through transcription and translation, it can happen at transcription. mRNA processing, so when we double check to make sure that the mRNA is good to go before we translate it. Moving and transporting the RNA from the nucleus to the ribosome and the cytoplasm, actual translation, protein processing, and then protein degradation or breakdown. So let's kind of go through each one. So we have DNA packing. Okay, how do you fit all that DNA into the nucleus? We've gone over this before. The DNA coils and folds around those histone proteins. Okay, it also helps that it's a double helix. Okay, so when you have that DNA double helix, you condense it to fit into the actual chromosome. Guys, my allergies are like super bad today. So then for our nucleosomes, those are basically your beads on a string. That's what they kind of look like here. So it's the first level of DNA packing. They're the histone proteins. It's a combination of 8 protein molecules that have positively charged amino acids. So that way they can bind very tightly to the negatively charged DNA, because remember we have those phosphate groups, so they're negatively charged. If we go back, you can see there's a little DNA packing movie that you can look up that's kind of about this if you need to see, I guess. So the degree of packing of DNA regulates your transcription, okay, because they're tightly wrapped around the histones, so there's no transcription. So that way the genes are turned off. If you can't transcribe them, the genes are turned off. Now we have this thing called heterochromatin, which is darker DNA. This is H, okay? It's very, very tightly packed. But then euchromatin is where the E is. That's the lighter DNA, and that's loosely packed. So the loosely packed ones are going to be the ones that are able to unleash and then transcribe. So all cells have their own pattern of histones and methyl tags. So methyl tags are just these little yellow dots that kind of tag certain parts of the DNA. DNA methylation blocks transcription factors, okay, which means there's no transcription. Here at the bottom, when I say methyl, you can see it's a CH3 group that's just kind of added on. Okay, so this methyl group is added to cytosine, so one of our nitrogenous bases. So that way it blocks transcription factors from coming in and starting transcription. So when there's no transcription, that means that your genes are turned off completely. Okay, so here it's just explaining they get the attachment of the methyl group to cytosine specifically. Okay, so this is basically almost a permanent inactivation of your genes. Once you have methylation occur on the DNA, then that's going to be pretty permanent of turning off your genes. Okay, so inactivated mammalian X chromosome, that's your X chromosome for like females, is where you have that bar body. Okay, so the bar bodies we talked about for specific disorders, for meiosis, okay, when we got that triple X chromosome or genotype. So one of the X's is inactivated, okay, and that's the bar body. So this is kind of to show you here we have the methylation, the methyl attached to the cytosine. So because of that, you can see here that it's still wrapped around, but it's just, it can't be transcribed. It goes to being wrapped around. So transcription initiation, controlling regions on the DNA. You have promoter, which is the nearby control sequence on the DNA. Okay, we went over this when we went over transcription. So the promoter is what kind of gives the signal of, hey, transcription factors, you can come on and start transcribing. So you have binding of RNA polymerase and transcription factors there. Now, this is the base rate of transcription. You have an enhancer, which has distant control sequences on DNA. All of these we went over for transcription. So this is where you have the binding of the activator proteins, and it has an enhanced rate or high level of transcription when this enhancer is bound. Okay, so this is kind of the model for the enhancer action. We're going over this because now this is turning on the gene. Okay, we're going to be transcribed. So the enhancer DNA sequences has the distant control, because here you can see the promoter, but then the enhancer is all the way at this end. So then you have the activator proteins that bind to the enhancer sequence, and that's what stimulates transcription. Then you have the silencer proteins. Those bind to the enhancer and block gene transcription. So if you silencer proteins binding there, that means transcription is not going to occur. So this is kind of the transcription complex. Looks real intense. Okay, so here you can see the yellow sites are the enhancer sites. Okay, you can see here the TATA box. So that way that's your promoter region, and then RNA polymerase attaches there. And then here's that coding region of the DNA that's going to start. These are your activator proteins, okay, that bind to the DNA at the enhancer sites to increase the rate of transcription. So this is what it looks like in real pretty complicated protein format. Now we can go into post-transcriptional control. So we went over RNA splicing when we went over transcription. So this is the alternative for the RNA splicing. Sorry guys. So you have variable processing of exons creating a family of proteins. Okay. Remember, our exons are the actually expressed parts, whereas the introns are just kind of the junk. So now, regulation of mRNA degradation. So the lifespan of mRNA determines the amount of protein synthesis. So mRNA itself can last from hours to weeks. So if a really big protein is going to be built, then it's going to last on the longer end. Whereas if it doesn't have that many exons, more introns, it's going to only last a few hours. So it just depends on how much protein actually has to be made from that piece of mRNA. So to control translation, you can block the initiation of the translation stage. So remember, in translation, the regulatory proteins are going to attach to the 5' end of mRNA. And so if you're blocking it, you want to prevent the attachment of ribosomal subunits and the initiator tRNA. So that way you can block the translation of mRNA to protein completely. So RNAi stands for RNA interference, and that's the complementary RNA that basically prevents and blocks translation stage. So now we are looking at how we can block or control genes in protein processing and degradation. So protein processing, that's literally just how the proteins fold into each other. Remember, they can have primary all the way to quaternary structures. So the cleaving, the adding sugar groups, targeting for transport, all those kinds of things. Then for the actual degradation, how are they going to be broken down? So there's this thing called ubiquitin, which basically tags the protein. It attaches to the protein, and then it tells the cell, hey, we need to break down the protein. So the protein is tagged, and then this thing called proteasome, sorry, I always have trouble with this word, proteasome, kind of wraps around it and breaks it down into small protein degraded pieces. So ubiquitin was founded by these dudes, and they basically called it the death tag. So this marks unwanted proteins with a label. So it tags them and is like, okay, they need to be taken out. So these labeled proteins are broken down rapidly in waste disposers, which are proteasomes. And ubiquitin itself is a pretty big protein. It's 76 amino acids. So this is kind of what it looks like. This is the cell's waste disposer. It breaks down any proteins into 7 to 9 amino acid fragments, and then the cell just kind of recycles them. Okay, so here's when we can have all the gene control. We have modification of DNA here, transcriptional, post-transcriptional, translational, post-translational. Okay, so I'm going to go into the summary and everything else in the next one.